I'm going to share a bit about the data management and how we do it in Leiden. And I hope that you give me some feedback on whether this is useful or not, and whether you can learn something from it. And I hope that I can learn something from you during this meeting, because I think we're all struggling to get this to work properly. That's also what I think data management is about. Right? What data management is about is basically changing this. Right? This is how it used to be. This is what we want it to be. But my users still usually prefer the way on the left, right? Because if you do like this, it's easier. Well, in the beginning, not when you have that much, but in the beginning it's easier. Walking around with a single hard disk is easier than sticking your data into a mirror, because you have to do more steps. So another problem with data management is this. There are many solutions. Omero is one of them. But before we started to have real data management at Leiden, our ICT service offered 17 different solutions on how to store your data, which is fun because everybody can do whatever they want. But it's not very useful because nobody knows where the data is. And even if you know where the data is, you don't know what happened to it. So I really love these comics, so I always use them. Together with NL Bioimaging, so we are 18 institutes in the Netherlands combined, trying to organize the Dutch bioimaging. So that's a bit like what you have here in Germany, but then the Dutch version of it. We're trying to elevate the bioimaging across the Netherlands, trying to bridge the biological implications and the technological ones. And of course, there's all kinds of analysis, AI-driven, self-steering, whatever. But the main reason for doing it is to be fair and to share these data in a way that you can actually reuse them, that they are retained for a long period, that you can see that the data was made properly, because we have quite some hiccups in the Netherlands with scientists that are cheating with their images and falsifying data, which we want to prevent. So you need to be able to look at it back, right? So that those things, you want to be careful with that, and you need to know where your data is coming from. So this is where we are. We're from Maastricht to Groningen and everywhere in between. As you can see, there are not 18 dots on the map. That's because some dots have multiple centers in the town. Amsterdam has 3, Leiden has 2, Delft has a few, Rotterdam has 1, Utrecht has 2, 3, I think. So that are multiple dots there. Each of these nodes are producing lots of imaging data, and we need to handle those, right? I'm going to focus a bit at what we do in Leiden, because we started doing this in the Netherlands. We're one of the front runners in this project. And that's why I can share what we're doing there and what is being implemented at most locations as well. So we combined different things in Leiden. We, of course, have an electronic lab journal. We use research space for that. We have Research Drive as our online storage for non-imaging data, and we have an Omera database running for online microscopy storage.
Yes. I have a question.
Yes, of course.
Research space the same as our space?
Yes.
Okay. Thanks.
Yeah. And we have our own MIPME model, which we integrated in these different data sets. Because we like the RMB model, but as you already said, it contains so many things to fill in that users get scared. So we decided to chop off a lot of it and actually make it more minimal, just to get people to used to it and then hopefully expand it in the future. Because we think starting with a smaller data set is still going to be useful. And then once you have them on board, you can slowly expand. That's hopefully that's what we are trying to do, at least. All this is done, what I'm going to show you in the Leiden Cell Observatory. We are 5 institutes that put all our microscopes together in one facility. They're all managed by the responsible institute. So I manage the biology instruments. There are people from toxicology that manage their instruments, people from chemistry that manage their instruments, but we can all share the instruments between each other as long as we talk to each other. They're in the same rooms, but they're still mainly used by the institutes themselves. We have around 60 microscopy systems, around 300 active users, and we've been using Omero since 2020. Last month, we passed the 5 millionth image that we have in our database, so we're happy with that. And they were created by about 620 users, and it's in total 130 terabytes, just to throw a few numbers at you. As said, all these things, we try to combine them, and that's also the thing. I think the main advantage of these things, both Research Space, Research Drive, and Omero create permalinks for their objects that you put in them. So what we try to do is actually should be able to access things from here. Let's see what happens. Yeah, go away. Let's see. Does it? Yeah, so here I go to my electronic lab journal, I go to my experiment, and then in that experiment, I actually have the links to my Omero and my Research Drive where I present this. So if I click this one, it will open my Omero, and it will show the image that I'm talking about and that I processed. Right? And the same thing, if I go back to if I have here in the comments, I have the attachments, both to this is my research space, or this is my data in Research Drive. And if I click that, then it opens the file in Research Drive, which shows the imaging that was done before. I actually fixed this for the SEM. It should open the PowerPoint directly. So you get to see the images based on the other microscope data, depending on how you have these things organized, but you can cross-link all of them together. And that's actually what we try and emphasize users to do, because it's very nice to have all the data in one place, but you usually can't because there's too much different things in there. So we emphasize on cross-linking all these connections so that it makes sense.
Sorry for interrupting.
Yeah.
So the link that you just clicked on from Omero side,
Yeah,
that was manually put there by
yes,
okay,
yes,
so it's
absolutely.
not like that
No.
gets pulled in from
No.
Research Space.
No, it's all manually put. Yeah, that's something that can be upgraded. We actually hope, and I'll show you in the next slide, that actually if this is what I already showed you, right? So from the lab journal, we can go to ELM. From Omero, we can go back. We add those links. And also here in the sheet, we can add links that you can link back. What we do want for Omero, if you're uploading the images, we use the MIPME model. We made that in an XML file, so you can use the endemic template matching that you have in Omero. I'm not sure that I have that here. I can show it later.
Yeah.
But then you can actually you can put the link already while you're uploading your data into Omero, and then it will get put as a key value pair, which makes more sense as a comment.
Then, sorry to just put on this, then just for the link or for the whole metadata, as a whole metadata description, so the long key value pairs in there.
We have the whole metadata in there. So we try to, we have a PhD course every year where which people have to follow when they start in their first year. We fill out the metadata template with them. They can save that. They attach it to the first directory in Omero. They can download it and open it, and they only have to adapt the things that they're changing in the experiment. Because filling it out every time is a hassle, and people don't want to do that. So that's how we did that. We changed our metadata template a bit. It's based on the ISA structure. So we have always investigation, study, and essay. And we have a file structure that is either in different tabs. So you can fill in the metadata either on your research drive where it's in an Excel file, or you can fill it in the ELN journal where it's a template, which is also structured in those three parts: investigation, study, essay. Or you can fill it in Omero where you actually get the connection directly if you're uploading the image with the endemic button that you have during the import, that you can click this model and open it from what you stored before. So there was 3 ways of doing it still, but it should be somewhere. And I really hate this bar on top, but okay.
Just click it away.
Can I click it away?
Okay.
Yeah.
And then under more, like meeting controls or like floating meeting controls. Yes.
Yeah.
That's
Okay. It's better. So because you want to link all these data, right? You want to have all the different metadata in there. There are too many models out there at the moment, which are all different. So that's why we think that sticking to one will not work. You can have REMBI for microscopy, you can have MIPME, you can have whatever, but you will need different ones for different types of experiments. And usually, most people aren't doing only imaging, they're doing other things as well. So you need to be able to connect them. And that's why in our version of our data, we think that actually having a map structure like we have in Research Drive helps us in structuring the data. And I'm not sure something similar is available in Germany, but. So what do we do with the metadata in Omero? I do actually have the example here. So I just added a picture, then we click the MDE button, and then if we select a picture, we can actually add the manual models that we made ourselves in an XML file. And you can click either the MIPME or the MixMe model, and you get to see this where you fill in the investigation, the study, and the assay details. Since we fill this out during our courses, it is easy for the user to reopen that data and just adapt it a bit, save a new version of it, and then reuse that again. So that's hopefully making it easier. Some people are convinced, some people definitely are not. So it will stay a struggle to get this to people to use this. It makes it easier for them at least. Same in ELN. In the ELN, we have this template where we just have all the different things below each other, which basically says the same thing. We don't have conversion tools yet. That's what we want to make so that you actually can make it in one of the three and automatically get it in all three, because that's, of course, even more useful. And in Research Drive, we just have the Excel file, right, which they can attach to their data. So that's how we see the three versions of adding their data. What we're doing now, we're trying to get a national Omero running in the Netherlands where we can actually share our data, because it's very nice that we have it all locally in every facility. There, I think now 5 or 6 of the 18 are running Omero, several are setting it up. But we need a national one where we can actually share data, which is not available yet. We're looking at integration also for the different resources. We're talking together with Bina to talk about metadata templates. They're actually talking about REMBI 2.0, right? So there's updates going on. Hopefully that will work. One of the things that's also developed in the Netherlands is BioOmero, which might be useful for you. It's a tool that connects your local Omero instance to your local high computing cluster, which means that you can drag or get data transferred from your Omero to your computing cluster, get calculations done, and send it back to Omero to connect your results there. If you don't need high computing cluster, we also developed a plugin for Fiji that downloads the data from Omero directly in Fiji, does a Fiji analysis, and then uploads the results back to your local image. So if you don't need a lot of power, that also might be an option. These are all the people in Allo Bioimaging, so there are a lot. The metadata and RDM people are on the left bottom, and we hope to get some serious imaging and metadata annotation done in the coming years with this money. Okay, thanks. That was all I had to show.
Are you happy with the key value nomenclature, naming structure from NDE? Or is it more of a, okay, we want the metadata in, this is a nice tool, but this is primarily for machine readability? Or are actually users looking at the table?
You can change the determinators yourself, right?
If
So-
you fit in Java and
So we rewrote the XML file ourselves.
yeah, the XML file, yeah,
Yeah.
but the nomenclature.
No, the people don't care how it's named. So they're happy with this. So as long as the data is in there, it links things together
Yeah.
that they can see.
Yeah.
Then they're happy with this. It doesn't matter what stands before that. They just ignore it. What they can't read, they just ignore is our experience.
So right now you were showing the template in MD that was empty, but when the people have their template, do they pre-fill with the pre-made value?
Yeah.
Each
Yeah.
of them have their.
Yeah, so during the course, we make them fill out one version of it. We save that, and while they're doing the course, they also upload some test images. We actually attach that template to that first test data, so they can, wherever they are, download it from Omero and directly open it again while importing.